This is a guide of how you can perform the ELISA test for the TB kit. It will give you all the information you need to do the test, including using Terra software. The kit boxes are labeled with the contents of the kit, the expiration date, and the kit's lot number. Allow all of the reagents being used to warm to room temperature, which is 18 to 25 degrees Celsius, for 30 minutes. Prepare all necessary equipment to perform the test. They are listed below. If the kit is brand new, reconstitute the freeze-dried conjugate 100X concentrate with 0.3 milliliters of deionized water. Mix gently to minimize frothing and ensure complete solubilization of the conjugate. If the kit is brand new, reconstitute the human interferon gamma kit standard with the volume of deionized water indicated on the label of the kit standard. This volume will change from kit to kit. Mix gently to minimize frothing. Please note, the newly reconstituted kit standard is viable for 3 months when stored at 2 to 8 degrees Celsius. Label 4 dilution tubes as S1, S2, S3, and S4. These tubes will serve as your standard curve for the assay. Pipette 150 microliters of specimen diluent into each of the 4 dilution tubes. Pipette 150 microliters of the interferon gamma kit standard into the dilution tube labeled Standard 1. Mix gently to minimize frothing. Pipette 50 microliters from Standard 1 into Standard 2. Mix gently to minimize frothing. With a new tip, pipette 50 microliters from Standard 2 into Standard 3. Mix gently to minimize frothing. No further dilutions are necessary for Standard 4. The dilution tube labeled Standard 4 will only contain 150 microliters of green diluent. Prepare the working strength conjugate in a 15 milliliter centrifuge tube according to Table 1 and the plate map. On the left is an example of a plate frame and wells prepared to run the assay. To the right is a plate setup representing the sample layout. In preparation of loading the microplate wells with working strength conjugate, first pour the conjugate into an unused reagent reservoir. This will allow for the use of a multi-channel pipette to simultaneously load up to 8 wells at once. Set a timer to 2 hours in preparation of the incubation period. Using a multi-channel pipette, draw up 50 microliters of working strength conjugate. Pipette the working strength conjugate into all wells. Using a multi-channel pipette, transfer 50 microliters of standards 1 through 4 into wells A1 through D1 and wells A2 through D2 respectively. Remember that the standards are run in duplicate. This is an example of standards 1 through 4 simultaneously pipetted into wells A1 through D1 respectively. Antigen tubes, which are the green and yellow tubes, are run with 2 levels of controls: the null control, which is the gray tube, and the mitogen control, which is the purple tube. These samples are pipetted into separate wells in the order of null, TB1, TB2, and mitogen. Using a single channel pipette, individually load 50 microliters of the null, TB1, TB2, and mitogen into their corresponding wells. Place the plate on a shaker and shake for 1 minute. Remove the plate from the shaker, place it on the bench top, and begin the incubation period by starting the timer that was set for 2 hours. While the plates are incubating, prepare the 1X TB wash buffer. Using a graduated cylinder, add 950 milliliters of deionized water and 50 milliliters of 20X concentrated wash buffer. Following the preparation of the 1X wash buffer, label the wash buffer as TB wash. Initial and label the buffer with the date it was created. When the 2-hour timer sounds, carefully flick the plate into the sink in a swift downward motion. Make sure the strips of wells are firmly placed in the plate frame. This will serve as a precautionary measurement to prevent the wells from falling out of the frame and into the sink. Pour the 1X TB wash solution into a clean reservoir. Set the multi-set pipette to number 5. This will load 250 microliters with each press of the lever. Insert 8 new pipette tips firmly. With the tips in the wash buffer, depress the multi-set pipette plunger to load the wash solution. Carefully pipette 250 microliters of 1X wash into each column. Lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid backing up into the pipette and contaminating it. Repeat this wash procedure 5 times for each plate for a total of 6 washes. Repeat this wash procedure 2 additional times for each plate for a total of 3 washes. Firmly and carefully tap each plate upside down into a paper towel. This is performed to remove as much excess wash as possible. Set a timer for 30 minutes. Use a new reservoir for the addition of the substrate solution. A full plate of wells will require the addition of 10 milliliters of substrate solution to a reagent reservoir. For 2 plates of wells, add 20 milliliters of substrate solution. Plan accordingly. Excess substrate solution may not be poured back into the original bottle. Use new pipette tips to load the substrate solution. Using a multi-channel pipette, draw up 100 microliters of the substrate solution. Transfer the substrate solution from the reservoir into all of the wells. Start the 30-minute timer as soon as the substrate solution has been loaded into the first column of wells. Try not to touch the edge of the wells with the pipette tips to avoid contamination. Never pour substrate solution remaining in the reagent reservoir back into the reagent bottle. This will contaminate the substrate in the reagent bottle. Simply discard any leftover substrate solution. This is an example of wells containing substrate solution following the designated 30-minute incubation period. Add stop reagent to the reservoir before the timer expires. Following the 30-minute incubation period, draw up 50 microliters of stop reagent into the pipette tips. Add the 50 microliters of stop reagent to all of the wells. Tap the side of the plates gently to mix the samples in the wells. This is an example of a completed TB assay. The standards are indicated. The rest of the stop reagent may be poured back into the reagent bottle. Place the developed plate into an ELISA reader for analysis. To read a new plate on the Terrace software, select File, Reading, New. Select TB-GP from the list of assays. Select Reader, Read and Collect. The reader will take 75 to 85 seconds to read the plate. This is an example of absorbance measurements displayed on Terrace for the TB assay. Cells A1 through D1 and A2 through D2 represent the duplicate standards 1 through 4 respectively. All cells that follow represent the null, TB1, TB2, and mitogen in alternating order. After measuring the absorbance values, select the Sample IDs tab. Select Edit On. Enter the sample IDs in the same order the samples were loaded into the microplate wells. Select Save once all sample IDs have been entered. A dialog box will immediately pop up. Type in the name of the test that is run, followed by the date and the initials of the individual who ran the test. Please see the example. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the TB assay, as well as a demonstration of test result storage within our proprietary software Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.